Good morning and good evening to you all. My name is Dr. Michael Risley, and I lead product development activities at Yourgene Health. I'll be talking today about Clarigene, a pandemic diagnostic story. To begin with, I'll give a brief overview of the content of this presentation. So I'll introduce Yourgene Health, including what we have to offer, our expertise and the company vision, and I'll move on to why and how Yourgene Health engaged in the COVID-19 diagnostic space, followed by the challenges involved in developing a COVID-19 diagnostic product during the height of the pandemic. I'll take you through design choices, covering the why and how decisions were made. And as Yourgene Health is in both the product and service business, I'll describe transferring the CE-IVD product into the service lab, and then go through the topic of SARS-CoV-2 variants and what this actually means for diagnostic products that are designed to detect this virus. So Yourgene Health is an international molecular diagnostics group which develops and commercializes genetic products and services. The group works in partnership with global leaders in DNA technology to advance diagnostic science. And primarily, we develop, manufacture, and commercialize molecular diagnostic solutions for reproductive health, precision medicine, and now infectious diseases. Flagship products include non-invasive prenatal tests, that's NIPT, the Downs syndrome and other genetic disorders, cystic fibrosis screening tests, invasive rapid aneuploidy tests, and a recent extension into the oncology space with DPYD genotyping. But at its core, Yourgene Health is about helping patients, from supporting diagnosis of disease as part of newborn screening to providing women with information to aid in the decision-making during pregnancy, and to helping prevent unnecessary deaths due to toxicity in cancer treatments. What we're really there to do is aim to enable scientific advances to positively impact human health. Going back just over a year, here in the UK, just as in much of the world, there were clear signs that we were moving quickly towards a national lockdown, and that could dramatically impact how the business would operate and function, with the potential for people to be unable to come into work. To prepare ourselves in the days preceding the national lockdown in the UK, we stress tested the business with a fire drill, and having only people who must be on site to actually come into the facilities. Fortunately, what we found was a great resilience, and we were able to operate very close to normal productivity. So when the lockdown hit, we were prepared. With Yourgene Health being in the medical device supply chain, it also meant that people who needed to be on site to manufacture products, for example, were eligible to do so. So as we were able to operate near normally across the business, across much of the business, and with this company vision in mind, aiming to positively impact human health, it meant that we were able and willing to involve ourselves in efforts to contain the virus. And at this point, while we had extensive experience in developing in vitro diagnostic products, infectious disease testing is not something that we had any real experience of. However, Yourgene's experience in the manufacture of those IVD products to the required quality standards meant that the product operations team could support the manufacturing scale-up of SARS-CoV-2 diagnostic products that have been designed and developed by other businesses. So we went into partnership and we utilized the product operations team's expertise and the facilities to make significant numbers of diagnostic tests on behalf of our partners. With supporting activities through this manufacture of third-party design and developed products progressing successfully, the next step was to look at whether Yourgene Health could and should develop a diagnostic product in-house. To decide on whether to do so, we performed a rapid evaluation with the aim of getting a handle on how the fast-moving situation was likely to play out. And with each passing day, the message seemed clearer that the long-term solution to getting out of the pandemic would be through the development of effective vaccines. But that would take some time, and until then, mass testing would be the key to allowing societies to exit from lockdowns and gain at least some degree of normality by identifying those who have been infected with the SARS-CoV-2 virus and enabling their isolation from the community to reduce the risk of onward spread. Of the different types of testing, Antibody testing was both outside of Yourgene's expertise and would only show whether people had been infected at some point in the past, while antigen tests were also not in our sweet spot. Plus, there was much debate around the accuracy and utility of this type of test at the time. Looking at the core technical expertise within the business and the likelihood that mass nucleic acid testing was a likely scenario for many countries, developing a product designed to detect the presence of viral nucleic acid seemed the obvious choice. And any business will always look to develop products in as short a time frame as possible to deliver these to market and help people, help the patients while also driving company growth. Developing a diagnostic during a pandemic really took this need for speed off the scale. Our standard process of entering an idea into a gateway funnel, gateway innovation funnel, building the business case, developing that deep understanding of the market via voice of customer analysis, as well as planning to resource and prioritize activities, went from a time frame of months to days. Fast tracking decisions and planning, we then pulled a team together from across departments to figure out how to quickly deliver a SARS-CoV-2 diagnostic quickly and effectively. With this cross-functional team, a new development needed to be pulled together without disrupting key activities, because we still had a number of key projects going on at the time. So the team had to be very smart. While we had quickly moved to remote working for most of the business, for the most part, this had allowed a continuation of existing activities that people were already familiar with, what would need to be done and what was coming next. For the new product development, this COVID test, the team needed to be formed from new while not being located together. So a new project team formed almost entirely over MS Teams. At this time, we also had people working at home with children and working very unusual hours, while others were able to access the lab and would be more working your typical hours that you'd expect, and somehow needed to blend this together and make it work. The product was also to be developed as a CIVD, so that meant absolutely no shortcuts allowable on any quality standards. So all the usual requirements for an IVD were to be followed, while at the same time aiming to reduce the time frame from development for a development project from perhaps up to a year down to just a few months. So we quickly discovered that expectations in regards to the supply of materials needed to be reset. With the pandemic being quite literally global, the whole world realized it would need these diagnostic tests. The diagnostic industry was itself continuing as usual to a greater or lesser degree, with an additional huge demand placed on materials for those used in relation to development and diagnostic use of SARS-CoV-2 products. The global supply chain for molecular diagnostics has a finite capacity, as with any other industry. So a consequence of putting these two factors together was a huge strain on the global supply chain, which would take lead times potentially from days or weeks to weeks or months. Enzymes, buffers, oligonucleotides, plastics were all in demand like we'd just never seen before. In the UK, the government also put out a call to develop diagnostic tests that didn't use the same supply chain. This led us to look at a range of suppliers and also to review raw materials we may not have otherwise looked at, such as perhaps a different or more bespoke selection of fluorophores. We also had to mitigate risks. So while being conscious not to add to the burden of the supply chain, such as ordering oligonucleotides from more than one source, but we needed to order and make sure we had quantities that were aligned to the project team's needs. Our procurement and logistics teams worked closely with existing and new suppliers to ensure we had just enough of what we needed and when we needed it. Reliable suppliers are key for any business, even more so at times when the supply was so constrained. As far as possible, a part of the strategy was to avoid using the same supply chains coming online via mass testing in the UK and elsewhere where we had visibility. To develop and validate a SARS-CoV-2 diagnostic also, as with any other diagnostic, also required access to clinical samples with known provenance. And it probably sounds a little strange given that a pandemic had just taken off with many people becoming infected, But in the early days of developing this product, this was a significant challenge. Few tests were available in early spring 2020, meaning that most COVID-19 cases were diagnosed with symptoms rather than a test designed to directly detect the virus. Of those in use, many were also homebrew with uncertain quality or unknown accuracy of results. So our clinical team made huge efforts to procure samples that we had sufficient confidence in to use as reference material for both test development and test validation. During the diagnostic development, Yourgene Service Lab also came online for routine SARS-CoV-2 testing. Initially, this used a third-party CIVD product. Where samples had the required consent, we could then use these to support Clarigene test development by accessing those eligible samples. When it came to selecting the type of technology, qPCR was the obvious choice, though an assessment was made early on as to whether we should utilize an alternative, either to avoid clashes with the existing supply chain or perhaps we could find some other beneficial reason. In the end, qPCR was becoming very quickly established and it didn't make sense to go against the tide. Plus, we also had considerable experience in developing qPCR products within the business. So this led into that particular technology choice. Basic features of the product also needed to be established at the outset. As a business with an international focus, a key question was to define a design that would be suitable for as many global territories as we could possibly achieve. Early on, the design requirements were not settled, with guidelines differing as to the number of targets required, whether specific targets needed to be included, or just with very few guidelines at all. The product design also needed to factor in technical needs and limitations. It seemed obvious that a product that had at least 2 viral targets was the right choice, because with just one target, a risk of false negative results was considered higher than when using multiple targets. Fast turnaround time was also an obvious need, along with just maintaining high overall accuracy. Having selected qPCR as the technology of choice, the range of platforms available across thousands of laboratories worldwide lent itself to a need to allow for compatibility with a wider range of these as possible. While Clarigene will be validated as part of the CIVD process on a defined instrument or instruments, we saw it as vital to allow for laboratories looking to use the product to be able to do so on any existing PCR platform available to them, particularly with the demand for qPCR instruments also seemingly exceeding capacity in a manner similar to the reagents and plastics demands on supply chain. A review was performed to understand which fluorophores would enable compatibility with the widest range of common platforms, and this pointed us to a set of fluorophores and using up to 3 color channels in total, covering viral detection as well as any controls we decided to include. The table here is an example which refers to a number of compatible platforms, though by no means all. The hazard of false negative results will be potentially very significant. Individuals could be given the go-ahead to interact with others, albeit in whatever socially distanced way is acceptable, meaning they could infect those they come into contact with. Therefore, we considered it that false negatives needed to be avoided at all costs. So reviewing the workflow, one obvious route for false negatives would be the failure of PCR across the whole plate. This is easily remedied with a positive control to be set up in each plate. If a positive run control fails, then the results must be discarded. Similarly, a no template control will be used to account for any potential false positive results. So an NTC amplification would result in discarding the plate. What the run controls don't account for is individual sample or reaction issues. Bad samples whereby RNA has degraded or a poorly set up individual well on a run with both valid run controls could still lead to false negative results. So the solution we came to was to utilize an internal control in every well, and one that requires the successful reverse transcription and amplification of a human RNA target. Therefore, any poor collections, mishandling of samples or errors in setup can therefore result in a failure to detect that internal control. The design of the internal control was therefore critical and should control for all steps in the process from sample collection through to analysis. Initially, we tested a CDC design, and this worked well. However, after reviewing the design closely, it was clear that genomic DNA could also be amplified by that particular primer and probe combination, meaning any contaminating DNA would also amplify, rendering the control ineffective for reverse transcription. So we put together a new design that would span the exon 1 and 2 boundary of the RPP30 gene and confirm the absence of amplification from genomic DNA, but also observing amplification from an RNA, a human RNA sample. With one of the 3 color channels used for the internal control, the assay design then allowed for 2 SARS-CoV-2 viral targets. This was considered to meet the need for reducing false negative risk, as 2 targets would require the unlikely sporadic failure of 2 amplifications, particularly as the positive internal control result would have to fail in combination. Looking at the targets to utilize, we both reviewed primer and probe designs available online, as well as looking at internally designed sequences. But the goal was not to design a completely unique and bespoke primer and probe design. It was to develop a high quality and effective product. So the selection was made on that basis. We selected designs of the targeted regions in the N and E genes. Of these, the N gene target was expected to be completely novel to SARS-CoV-2, while the E gene target is an area conserved from the original SARS virus. This absence of specificity of one of the targets to the SARS virus was not considered likely to present any challenges due to SARS no longer being in circulation. While the analysis could itself be tuned to allow for detection of both targets as required to give a positive result. So utilizing a conserved region was also considered to present a lower risk of mutations occurring that would render the design ineffective at detecting SARS-CoV-2. And as we have seen since then, products detecting targets in the genes encoding the spike proteins have faced challenges due to apparently higher rates of mutations in these regions. With that assay design in place, the workflow and analysis was the next item to lock down. So to talk you through the product and the assay, the setup involves RNA extraction from a sample arriving in the lab, typically a nasopharyngeal swab, before it then moves on to qRT-PCR setup. Each sample and control is set up in a single reaction well, and the samples and run controls are all then placed on the plate onto a qPCR instrument for just over an hour before the output file is available for analysis. The analysis itself is straightforward and uses CT cutoffs, so the positive and negative control must be positive or the whole run fails, or valid or the whole run fails. Samples negative for SARS-CoV-2 must give an internal control result to be considered as valid and have confidence in the result. Both SARS-CoV-2 signals are required for a positive call, and a single SARS-CoV-2 signal is considered as an indeterminate result, whether that's with or without the internal control amplifying, and then no signals from anything is considered invalid. So with the assay design workflow and analysis defined, the team went on to optimize the reaction and reagent conditions, kind of typical optimization robustness studies, and then we got to a final unlocked product. So the next step from there was then to demonstrate the performance of the test across a range of design verification and validation studies as required for a CE-IVD product. Focusing here on 2 of these studies, data of particular interest was considered to be the limit of detection and clinical performance. Across multiple products for SARS-CoV-2 detection, diagnostic detection, there are a range of LOD claims, quite a wide range in some cases, with a range of methods also used to generate the claims. One of the things that has been challenging is the absence of a consistent standard means that LOD claims are not simply defined by the product, but they are also defined by the sample used to generate the data and the claims. So for example, an overestimate or an underestimate of the copies per volume, per microliter. For instance, content from a standards manufacturer would in turn then skew LOD claims for the product that is using it. Time and care was taken by the Clarigene development team to obtain a reliable standard whereby we had confidence in the number of copies per microliter. And ultimately, we were able to demonstrate quite a low LOD successfully of 5 viral copies per individual qPCR reaction. Clinical performance was assessed using a set of samples with gold standard reference data. Of these, 100% detection was observed with no false positive results. To support the in-house study, an independent assessment of the Clarigene product was also performed, and this also showed 100% detection of positive cases with a very low false positive rate. The gold standard itself was another SARS-CoV-2 diagnostic, so the false positive results, the few that we saw, could have either been true false positives or simply that Clarigene is a more sensitive assay and detected real positives at low concentration. But the data here is presented as we see it, and it shows excellent results. Successful design validation alongside an effective transfer to product operations ensured that the CE-IVD status for the product was achieved at a fast pace. With the Clarigene SARS-CoV-2 IVD assay CE marked, the next step was to get it in use, supporting the fight against the pandemic. The obvious rollout was to Yourgene Health Service Lab in Manchester, and this provided an immediate impact, initially with private partnerships that enable people to go into their workplaces in the knowledge that they and their colleagues were COVID-free. Implementing the test in the service lab enabled us to quickly see that the launch test was reliable and robust in a high throughput environment. The rollout was efficient, with the lab able to use the Clarigene product in place of a third-party test that they had been using prior to Clarigene CE marking. They were also able to automate the PCR setup process in a short time frame, and in partnership with the software development team at Yourgene, developed software that was there to manage the testing process and tracking of sample information, which was also developed and implemented. In combination, this delivered a highly effective, automated and scalable SARS-CoV-2 diagnostic testing system in our laboratories, which could then be potentially rolled out elsewhere. So within a few months of launching the COVID-19 diagnostic testing service, the lab was also approved for several UK national testing purposes, including fit to fly, test to release, and also a sequencing service for viral surveillance, demonstrating the high quality of both the SARS-CoV-2 test as well as that of the service lab in Manchester. But designing, validating, and launching a SARS-CoV-2 product is not the end of the story. All CE-IVD products require post-market surveillance, though where Clarigene differed from Yourgene's and many other products is the rapid evolution of the virus, leading to mutations that seemingly randomly appear throughout the genome. As mutations arise, they could appear in a location that degrades product performance and results in a primer-probe combination with reduced ability to amplify in strains with that particular mutation or mutations, or lose the ability to amplify completely. The method of determining which strains and mutations cause risk to monitor databases such as NCBI to determine the location of mutations detected in the viral strains. From there, any mutations found to be located within Clarigene primer or probe locations will be tested in the laboratory. A fast and simple test is performed whereby DNA template containing the mutation or mutations will be tested alongside a wild type template sequence and the CT values compared. So as an example here, we have tested a mutation and showing an amplification plot. So this is for showing mean CT values for wild type and S235F variants of the B117 lineage, which is often referred to as the UK variant. Testing was performed for wild type and mutant S235 samples at 2 different simulated viral loads that 100 times LOD and 10 times LOD. The CT values are virtually identical, and this illustrates that there is no significant difference in performance for the Clarigene assay with either the wild type or the S235 sequences, with both detection rate of 100% in both the different LOD values. It's possible at any test at some point a strain will emerge where the mutation does impact the assay. Many such mutations have been found across the SARS-CoV-2 genome, and other tests in the market have found certain strains problematic, highlighting this need for constant monitoring. So hence why we see it as also important that we have backup prime and probe combinations and can plan to implement these into the product should the need ever arise. The regular monitoring of SARS-CoV-2 mutations supports making this feasible, as it improves the chance that we would identify any such problematic mutations before that strain takes hold in a population. So this takes us through where we are today. So what have we learned? That developing and ensuring the supply of materials for a diagnostic assay rapidly during a pandemic is very challenging. It's getting the key design decisions right early on is key focus, as is getting the supply chain right. So always key concerns, but even more so during this time. What we've achieved, we have a robust diagnostic product on the market, which has secure supplies of materials for high volume manufacture. And where do we go in the future? Well, that's something that we do regularly review, considering the SARS-CoV-2 market and where that will go, as well as now we've learned a little about infectious diseases, perhaps further involvement in the diagnosis of other infectious diseases. That brings me to the end of this presentation. Before I finish, I'd like to give my thanks to the Yourgene team involved in Clarigene in all capacities. It's been a huge effort from everyone involved. And I'd also like to thank you all for taking the time today to listen to this presentation. I hope you found it valuable, and I'm grateful for the opportunity to talk about Clarigene's pandemic diagnostic story.